Hi everyone and welcome to this presentation. I'll briefly introduce myself. I'm Gaurav Agrawal. I'm the Scientific Development Manager at Eurofins DiscoverX. Here I support scientific integration of our cell-based assay platform in drug discovery programs and lot release testing globally. So thank you for joining me today. In this presentation, I'll be focusing on how our qualified cell-based assays can accelerate the development and market release of COVID-19 therapeutics that are targeting the pro-inflammatory cytokines. So let me start with a brief introduction of our company. So here is a snapshot highlighting our core capabilities. Eurofins DiscoverX is the products brand of Eurofins Discovery, and we are based in San Francisco Bay Area. We are enabling drug discovery and development programs for over 20 years now. We come from a deep experience in engineering novel MOA reflective functional cell-based assays. We have the largest menu of cell-based assays covering 10 plus major target classes in phase-appropriate fit-for-purpose formats such as qualified ready-to-use bioassays, stable cell lines, and optimized assay reagents. We are leading the industry in partnering with biopharma industry and implementing our assays in their drug discovery and QC lot release programs. We are also closely working with their affiliated CROs and CDMO sites, ensuring successful method transfer and assay implementation in timely manner. Here is a bird's eye view of several such programs where our qualified bioassays are implemented in potency, stability, and NAB testing programs across the globe. We are highly committed to providing end-to-end support to such biologics development programs and ensure successful implementation of our fit-for-purpose qualified bioassays for validation at GLP GMP labs. Now, utilizing our core enzyme fragment complementation technology that I'll discuss in the following slides, we have built, in fact, the largest menu of cell-based assay portfolio in the industry with over 800 functional cell lines, and from that, hundreds of assay-ready kits and membrane preps. Our portfolio sufficiently covers all major druggable target classes such as GPCRs, receptor and cytosolic tyrosine kinases, interleukins, checkpoint modulators, as well as reporter assays for key regulators of signaling pathways such as NF-kappa B, STAT, and NFAT. And also we have assays for ADCC, ADCP, and other cytotoxicity applications. Now in parallel, we have been building ready-to-use bioassay formats for major targets and are qualifying them with clinically relevant drugs so they come pre-validated, thereby de-risking their implementation and lot release programs. So let's dive right into our focus area and efforts for accelerating COVID-19 therapeutic drug discovery and release programs. With nearly 150 million global cases and over 3 million deaths, COVID-19 pandemic has challenged the capacities of healthcare systems worldwide. From the pathology of COVID-19 in severe cases, it is apparent that patient's immune system mounts a highly disproportionate and aggressive inflammatory immune response, actually releasing several pro-inflammatory cytokines towards the virus. This is often termed as cytokine storm. Due to this, patients will be prone to getting secondary infections and multiple organ failures, which may lead to death. Some of the key pro-inflammatory cytokines I have listed here are interleukin 1, interleukin 6, TNF alpha, GM-CSF, and some of the chemokines. And as a result, there are several ongoing clinical programs that are evaluating therapeutics that are targeting inflammatory signals as a treatment strategy for COVID-19. As expected, most of these trials are focusing on repurposing the drugs that are already approved for other conditions. Here I've listed key cytokines and chemokines that are mainly implicated in COVID-19 cytokine storm and with their respective therapeutic drugs that are currently in clinical trials. such as tocilizumab targeting interleukin 6, anakinra targeting interleukin 1, adalimumab, that is neutralizing antibody for TNF alpha, and each of these drugs are part of several clinical trials across the globe. The figure here mostly lists the repurposed drugs, but there are also many new therapeutics that are targeting GM-CSF and IL-8 that are also being explored as a treatment strategy for COVID-19. Now, for development and characterization of these therapeutics as well as releasing them into the clinic, reliable functional assays are required. Now, this is where Eurofins DiscoverX can partner with you with a reliable cell-based assay portfolio. As you see here, we have functional cell-based assays for each of these cytokines and chemokines. Now, these assays can really accelerate the drug development and release programs, and how they accomplish that would be in the following part of the presentation. What you see here is a typical roadmap for developing a fit-for-purpose bioassay for a biologic drug candidate, which is needed to release it into the market for clinical use. It can take upwards of 12 months if starting from cell line development, subsequent qualification, and at the end implementation for QC lot release. And here is where our qualified pre-validated bioassays can really help in shrinking this long time frame by over 9 months. When implementing our bioassays which are qualified with the drug candidate, you will be able to bypass a number of milestones and can be looking at a ready assay that can be directly transferred to a GMP lab for assay validation work. This way, our bioassays can significantly accelerate time to market for the therapeutic drug candidate and help save significant costs and resources. So in this part of the presentation, I'll introduce you to our cell-based assay platform and present a case study for tocilizumab bioassay, which is designed for potency and stability testing for therapeutics targeting interleukin-6. And with that, I'll give you a quick overview of 3 other bioassays, which are for anakinra, GM-CSF, and TNF-alpha, which are other key cytokines that are also implicated in COVID-19 cytokine storm. As you see, these bioassays have been qualified with the marketed drugs, which makes them highly relevant for biosimilar and biobetter drug release programs. Now, following this, I will discuss our signaling reporter assays, which provide a more downstream readout. And towards the end, I will tie it with another set of assays for characterizing small molecule drug candidates and discuss functional assays for evaluating JAK1, 2, and 3 inhibitors. But let me first briefly introduce our enzyme fragment complementation, or EFC, which is our core technology behind these assays. Our assays utilizes this proprietary technology, which is based on split beta-gal system, where the enzyme is split into 2 inactive fragments called ED, or enzyme donor, and the EA, or enzyme acceptor. When these 2 fragments are put together, they complement each other to form a functional beta-gal enzyme. We add a chemiluminescent substrate in the assay that this active functional enzyme hydrolyzes to produce a light signal, and that can be read with any benchtop luminometer found in most labs. Now let me quickly show you how we use the EFC technology with an example of a receptor dimerization assay that we created for interleukin-1 receptor with its co-receptor interleukin-1 receptor associated protein. These receptor units dimerizes in presence of interleukin-1, which also represents its physiological MOA. As you see here, these receptor units are tagged with the beta-gal fragments as I described on the previous slide. Now, in the presence of interleukin-1, the receptor units dimerizes, thus bringing together the beta-gal fragments, and addition of substrate produces a quantitative gain of signal response as depicted below. Now, this assay can also be used to quantify inhibitory responses from an anti-receptor or an anti-ligand antagonist, which also produces a quantitative dose response curve. And likewise, we have successfully utilized EFC technology for many other assay formats, such as quantitating protein-protein interaction, protein turnover and degradation, for evaluating downstream response, and more recently for developing cytotoxicity assays such as ADCC and ADCP. Now for our cell-based assays, we offer phase-appropriate assay formats. One is of course the stable cell lines, offering a continuous culture format. But more importantly, we have the bioassays, which are our ready-to-use format and don't require any cell culture. It is our highly optimized assay format that is developed for potency and stability testing and are perfect for implementing in QC lot release programs. We took a step forward and have also qualified our bioassays with marketed drug where available. Now before I move forward, let me give you a very quick overview of a typical assay protocol for our cell-based assays. It starts with plating cells in the assay plate, usually overnight. Next day, the molecule of interest is added and incubated for another 1 to 3 hours. Thereafter, you will add a detection reagent with the beta-gal substrate in it and incubate the plate in dark for an hour to 3 hours. And that is about it. After the incubation, the plate can be read in any benchtop luminometer to obtain a quantitative signal. The schematic on the lower right side here shows a bioassay kit that comes with all necessary reagents included, tried and tested, along with the certificate of analysis document that comes with each kit, and it is lot specific. Perfect. I think we are ready to review a case study for one of the qualified bioassays. I have selected our tocilizumab bioassay since it is for interleukin 6 targeting therapeutics and thus most relevant for COVID-19. The intent of this study is to provide you with an overview on how we go about qualifying our bioassays to make them more suitable for potency and stability testing for drug candidates when implemented in a lot release program. Now let's look at the assay design for this bioassay. Of course, it's based on our EFC technology and utilizes the dimerization platform, very much like the interleukin 1 assay that we reviewed a few slides above. Now in presence of interleukin 6, the receptor units dimerizes, bringing the receptor units together and thereby complementing the EFC fragments. Now adding of the beta-gal substrate produces a quantitative signal. However, when the assay is run in the presence of tocilizumab, which is an IL-6 receptor antibody, the binding of IL-6 is inhibited in a dose-dependent manner and also produces a quantitative inhibition curve. Now here are some of the key assay parameters we evaluate during assay qualification. First, we evaluate assay consistency between several dose response curves. This gives a very good idea on the assay variability associated with the assay. Second, we look at plate uniformity across the entire plate, and this provides us with any plate associated bias and possible edge effects. Next, we look at assay repeatability and for consistency in the slope of the curve. And lastly, we also perform assay suitability for relative potency analysis. We determine assay accuracy, precision, and linearity over a range of 50 to 150%. And this is performed by 2 individual operators. So our assay qualification, as you can see, is based on ICH guidelines. Here is the data for evaluating intraday plate-to-plate variability. 3 experimental replicates of tocilizumab were prepared at 11-point dose response curves and run on the same plate, with duplicate wells per dose on the same day by 1 operator. Similar experiments were repeated again to obtain data from 3 individual days for intraday variability. As you can see, the curves were very uniform in these experiments. And next slide, we analyze the data statistically for intraday and interday reproducibility. The table on the left shows analysis of each curve with excellent interplate repeatability and intermediate precision of both assay window and IC50. The precision was within 8% for the signal to background and below 10% for IC50. This is an excellent result for experiments that are run on the same day in separate plates. Now, the table on the right here shows a culmination of 3 such experiments to calculate intraday precision. As you can see, it is below 25%, which is again well within our acceptability criteria. All the data here suggest an excellent precision between multiple set of experiments run across multiple days, and this is something that's required when the assay is implemented in a quality controlled environment for drug release. Next, we performed relative potency experiments and parallel line analysis with tocilizumab across a range of 50 to 150%. The experiments here show data from two individual analysts. The blue curve is with 100% reference, while the green is a sample. As we move from 50% to 150%, you can see the green curve shifts towards the left and move across 100% reference, depicting an increase in potency. Here's a statistical analysis of the relative potency data. The assay was determined to be very accurate with 105%, and overall assay inaccuracy was within 9.3%. A high degree of accuracy, as we understand, suggests that the assay can measure relative potency with high confidence. On the other hand, assay precision was below 6%. Now, such high degree of precision here shows an excellent assay repeatability when performed on different days and between different analysts. Dilution linearity was analyzed plotting the measured versus the expected relative potencies as shown in the figure to the right. The R-square values just came below 99.3%, showing an exceptional assay linearity. Now, these data sets that I just discussed for tocilizumab bioassay is a part of our standard assay qualification work for all our bioassays. Now, with such amount of assay qualification work behind them, you can be confident when implementing them in your quality control space for biologic or biosimilar drug candidate. Next, I will discuss three other bioassays briefly and not go over the entire qualification work, but please feel free to reach out if you would like to review them. Here are three more bioassays also qualified following the ICH guidelines. These are dilutional linearity data derived from similar relative potency experiments I just described for tocilizumab bioassay. On the left, it is the adalimumab bioassay for TNF-alpha qualified with Humira. In the middle, the anakinra bioassay, which is qualified by Kinret, and on the right, the sargamestim bioassay, which is qualified with Leukin. And as you see, all these bioassays, just like the tocilizumab bioassay, also presents excellent linear potency with clinically relevant drugs. Next, I would like to introduce our new set of reporter assays designed to provide a more downstream readout for the activation or inhibition of a signaling pathway. These would be highly complementary to our bioassays that are more focused on recording upstream events. I will show you three different reporter assays for cytokines that are relevant to COVID-19. The first is the HepG2 cell background exploiting the STAT3 pathway, whose role is well documented for cytokine signaling, including interleukin 6. The figure here shows a robust and sensitive response to interleukin 6 and a good inhibition curve for tocilizumab with a large signal-to-assay background window for both. Next is our NF-kappa B reporter assay in U2S cells for TNF-alpha. TNF-alpha, like many of its family members, signals through NF-kappa B pathway. The figure here shows a robust and sensitive response to TNF-alpha and a good inhibition curve for Humira with a very large over 50-fold assay window for both these molecules. The other assay I would like to mention here is the rank receptor assay. So rank is a member of TNF family and binds to TNF superfamily receptor 11 and has a role in tissue growth and immune functions within the body. This reporter assay overexpresses the rank receptor in the U2S cell background, which also utilizes the NF-kappa B pathway. The figure here shows a highly sensitive rank L dose response curve with over 24 assay window and a good inhibition curve for Prolia. Prolia is the marketed drug for denosumab, which is an anti-rank L antibody. Thus, combined with the bioassays, these signaling reporter assays will provide a comprehensive understanding for the drug molecule's MOA. In this last section, I would like to highlight our functional cell-based assays for JAK inhibitors. JAK inhibitors have become an area of active investigation with several ongoing clinical trials for COVID-19. The data on the right is a functional assay where JAK1 is expressed as a cytosolic tyrosine kinase along with CSF3 receptor. When GCSF is added, CSF3 receptor is activated and is phosphorylated by JAK1. Similarly, the assay in the middle expresses TYK2 with growth hormone receptor and JAK3 with prolactin receptor in the JAK3 assay, which is on the left. Now together in these assays, we have tested several JAK inhibitors, and most relevant among them are shown here, such as ruxolitinib itself. I'll stop here and would like to quickly mention that we have a strong custom assay development team who can build assays or modify existing ones to suit your needs. We can also take up pilot studies if you would like to run a quick feasibility study with any of our assays. Plus, our Eurofins Discovery Partner Labs can run any of our assays as a full service for you. So, a quick set of conclusions. In this talk, we have discussed our target-specific, qualified, MOA-reflective functional assays for drugs targeting the pro-inflammatory cytokines, all of which are implicated in COVID-19 pathology. With that, we offer complete solutions, including custom assay development service. One relevant example is an ongoing custom assay development project for ACE2 receptor that is intended for characterizing therapeutics that are aimed at preventing the SARS-CoV-2 virus from attaching to the alveolar cells in the lungs. For more information, please visit our website at discoverx.com. Revisiting the slide from earlier in the presentation, we have shown here how our qualified pre-validated bioassays from Eurofins DiscoverX can really accelerate drug release programs, mainly because most of the time-consuming steps in the assay development and optimization process are already accomplished. In addition, our assays have undergone qualification with the innovator marketed drugs based on ICH guidelines. As a result, the time frame for assay development in GMP labs can be reduced by over 9 months, therefore significantly saving on time and project cost. With that, I would like to thank you for joining me today.